All right, so this is going to give you the basics of what Hardy-Weinberg equilibrium is, what's the mathematical model, and just the basics of it. In class, we'll do more problems. I'm going to do an example problem at the end of this, but in class, we're going to do more problems and get a better handle on how to do different types of Hardy-Weinberg problems. So Hardy-Weinberg was developed by two different people. That's why it's called Hardy and Weinberg. Hardy was an English mathematician. Weinberg was a German physicist. And I think they actually developed these theories in the equation independently, but they're both credited. That's why they're both credited for the idea and discovery. All right, so the Hardy-Weinberg principle. First is we are going to look at mathematical representation of essentially evolution in a population. And so our first equation is P plus Q equals 1. The P and the Q are frequencies of alleles in a population. And in this instance, we're looking at two alleles for the same gene. The larger equation, P squared plus 2PQ plus Q squared, is looking at the frequency of the different genotypes, because in a diploid organism, you're going to have two of each allele. So P squared is the homozygous dominant, 2PQ is the heterozygotes, and Q squared is the homozygous recessive. All right, so we're going to use this then to predict genotype frequencies within a population. And these frequencies of alleles will remain constant from generation to generation if they are in Hardy-Weinberg equilibrium. Essentially, if evolution is not occurring on a trait, if the trait is not changing within a population, then it's in Hardy-Weinberg equilibrium. If this is not the case, then evolution is occurring. We are seeing a change in allele frequencies, and that's all the mathematical model is testing. So remember, our definition is evolution is a change in allele frequencies over generations within a population. And that's what this equation then tests for. So let's start with our main assumptions. We first are in this equation. You can expand this equation, but we're just going to keep it simple. In a population where you have two alleles for a gene at one locus, so one location at this gene, and then you have two alleles. P is the frequency of allele 1. And so we're making a decimal at this point. We're going to take the total number of alleles, and we'll take the total number of allele 1 to determine then the frequency of allele 1. So it's going to be something between 0 and 1. So it's going to be a decimal. Q then is the frequency of allele 2. So we've done a lot of these Punnett squares where we have two alleles and they account for different traits. There's a whole list of them, of the ones that Gregor Mendel used for the different traits in pea plants. And each of those are great to use because we got two alleles that control one trait, and that's all that's in the population. Okay, so that's P and Q. So an example, if there are 100 plants with two colors of flowers, which are determined by the presence of two alleles at one locus, and these are the way it breaks down. A homozygous dominant is big R, so allele 1 is dominant to allele 2. And our heterozygous then is also red, and then our homozygous recessive is white. So what is P and Q if 50 plants are big R, 20 are big R, little r, and 30 are little r, little r? Well, all we're going to do is we're going to count all the alleles. We're not going to count the individuals, the alleles. So if there are 50 big Rs, then we do 50 times 2, because that's how many big Rs are in our population. If there are 20 big R, little r's, then there are 20 R's in that population, big R's, and then 20 little r's. So we take that, we multiply it, and then we divide it, we add them together, and then we divide it by the total number of alleles, and that's where we get 0.6. Okay, we do the same thing for Q with our recessive allele. We've got 30 homozygous recessive individuals, 30 times 2, and then we add the 20 little r's that are in our heterozygotes, and you get 0.4. P and Q will always add up to 1. So if you get two numbers that don't add up to 1, you either maybe rounded the numbers differently, or you did it wrong. Okay, so that's one way to check if you did at least this part right. All right, again, so now we're going to divide up into our different traits, and we take the square of p plus q. So p plus q squared gets you p squared plus 2pq plus q squared, and you can do the FOIL method if you want to work that out. So in our example, we had 0.6 and 0.4. These frequencies, our p and q, can then be used to predict the frequency of each genotype if mating is random. So the probability of big R is p squared. The probability of little r is q squared. And then the probability of big R is 2 times p times q. And so that's where we get these numbers. 0.48 is for heterozygotes, 0.36 for homozygous dominant, and 0.16 for homozygous recessive. All right, now these are decimals. And so if we were to then look at the expected frequencies, we would multiply each of these by 100. Because in our population, we have 100 individuals. 50 plus 20 plus 30 is 100. And so we take our expected frequencies, which we calculated from our first equation, p plus q equals 1, and then p squared plus 2pq plus q squared. And then we're going to compare that to our actual frequencies. So is this population in a Hardy-Weinberg equilibrium is the question. Well, let's look at what we actually have versus what we would expect. And when you look at that, you can see that these numbers are not exact. Now, if you wanted statistics to look at the probability of getting these results, we could run a statistical test. But we're not going to do that in this class. We're just going to kind of eyeball it. So our numbers are off. 50 to 36, 20 to 48, and 30 to 16. That's much different than what we got. So we would say that it is not in Hardy-Weinberg equilibrium because those numbers don't match up. All right, well, why would a population not be in Hardy-Weinberg equilibrium? Well, that could be any of these 5 reasons. So these are the assumptions of the Hardy-Weinberg principle. So if a population is in Hardy-Weinberg equilibrium, that assumes that there are no new mutations in this trait. There's random mating, there's no natural selection, that's a very large population size, and you don't have individuals coming in or leaving the population, which would be emigration or immigration. We can also call that gene flow. So we would say, well, looking at our flowers, perhaps there is a preference by bees towards a color, and maybe that would account for our difference in our expected and actual frequencies. And so we would say, well, probably the random mating is the one that's being violated. We would then have to do more data and research to check and see if that's true, but that would be then a place we could possibly start. If any of these, however, are not met, we would say the population is not in Hardy-Weinberg equilibrium, and that then the allele frequencies are changing, thus fulfilling our definition of evolution. We would say this population is evolving. Or another way of saying that is if a population does not exhibit Hardy-Weinberg equilibrium, then it is likely evolving due to one of these 5 factors. Okay? All right, so let's give you another example. Phenylketonuria is a rare genetic condition where individuals cannot metabolize an amino acid called phenylalanine, which is found in meat and dairy, eggs, nuts, products, high protein foods. It's also a principal component of aspartame. So when aspartame breaks down in your body, phenylalanine is one of the components. And so people with phenylketonuria cannot eat those foods. It's a recessive disorder found in about 1 in every 10,000 babies. Okay, so that means you have to be homozygous recessive in order to actually show the disease. But lots of people can be carriers of the gene of the condition, but not actually have PKU. So, using our current medical practices, we can assume, or we are assuming, that it meets the 5 assumptions for Hardy-Weinberg equilibrium. And so the question we can ask then, all right, well, what percentage of the population then is the carrier of the gene? So to answer this question, we have to think, all right, well, what information over here? We've got P plus Q equals 1, we've got 1 in 10,000. What information here correlates with our equations? Well, we know that to have PKU, you have to be homozygous recessive. Okay, so 1 in 10,000 is actually our Q squared. All right, so 1 in 10,000 people, that means that one person has 2 alleles, and the population would have 20,000 alleles. So to get Q squared, we can just divide 2 divided by 20,000, same as 1 divided by 10,000, which is 0.0001. All right, so now that we have Q squared, we can actually determine what Q is just by taking the square root. So we take the square root of Q squared, and we get 0.01. And what are we interested in? We're interested in knowing the carriers. The carriers are heterozygotes. Okay, they have one of allele 1 and one of allele 2. And that is represented in our population by the 2pq. So if we can calculate 2pq, we can calculate the answer. So we need to know p, and we have q. Well, we know that p plus q equals 1, so we can plug that into our first equation to get p. So p is 0.99. Now we have a value for p and a value for q. All we need to do is calculate 2pq. 2 times p times q is 0.0198. Our answer then is 1.98% of the population are carriers of PKU. So this is just one of the type of questions that you can get. We'll spend a lot of time practicing these, and you'll have one of these on the exam as well.